Okay guys, so in front of you, you should have the notes that look like this, except with a lot more blanks because you have the fill in the blanks version, which you will be writing on as I am going over these notes. So I'll be reading over these notes and highlighting what you should be writing on your notes. So also remember that there is no textbook for this class, so everything that could possibly ever be on a quiz or a test or anything we talk about in class, you're going to want to reference these notes. So that means that you should be taking the notes as I'm writing them, since I'm giving you class time to do it. And you should also be actively listening. So not only you're writing, you're also listening to what I'm saying and be thinking about comparing that to things that you already know. Okay? So we're going to start here on the scientific method, which is something that you are probably familiar with. You've heard it before, hopefully, but we're going to still talk about it a little for this class. So science is a study based on observation and data. Based on observation and data utilizing the scientific method. So remember, in science, everything is based on observation, is based on collected data. So it's not just a guess. Everything in science that we believe is a fact backed up by lots and lots of data, okay? Science, like we said, deals with data, not with assumptions or ethical problems. So in science, we have to be careful with making assumptions. If we see something, if we observe something while we're experimenting, it might be easy to make an assumption or a guess, an educated guess, because that's what you've been told to do in the past, that's what you're used to. But in an experiment, you should never assume anything. Anything that you conclude should be based on data. And science also does not deal with ethical problems. So scientists aren't worried about whether humans or animals or anything should do something. They look at the causes and the effects if, say, humans do something. So should humans litter? They're not worried about that. They look at the causes and the effects of when humans do litter. But you shouldn't litter. And then this third bullet here, the scientific method is a logical, organized way of studying a problem. And because it's a logical way of studying a problem, being a scientist is universal. So anyone can be a scientist if you think in this logical, organized manner. Okay, so next we will move over to the steps of the scientific method, and we're actually going to do this together as a class. So if you're to this point where you need to be filling this part out, pause the video, take out your headphones so I know that you're ready to move on with the class. So now that you should have this part filled out that we did in class together, we are going to be talking about the parts of an experimental design. So one of the biggest parts of the scientific method, one of the biggest parts of science is experimenting. And there are a couple key parts of an experimental design that we're going to talk about now. So first is the hypothesis, which hopefully, again, we've heard of, but we're going to talk about it a bit more for what I think fits this class. So the hypothesis is a statement that can be tested. It's a statement that can be tested, and here it's an educated guess. So you're not expected to know what is going to happen, because why are you running the experiment if you already know it's going to happen? In an experiment, you're running it to figure out things that you don't already know. So you have some information on the topic because, hey, you did the background research over here. So you know kind of what should happen, what you think is going to happen. You're smart. You think something's going to happen, and that is what your hypothesis basically is. It is a guess. It is a prediction as to what will happen. And a hypothesis is different from just any old guess because it's written in a very specific format, okay? And this is where I think maybe it might be new to you, or you might not have explicitly heard this part, so pay attention here. Where this third bullet is, it says the hypothesis is written as an if-then statement. And you'll hear me talk about if-then statements, and that is what I mean. I'm literally telling you the format I want it in. I want it to start with if. It'll say if blah, 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 then blah, 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 blah. And we'll talk about examples of good hypotheses, and you'll be coming up with your own hypotheses for different things. Plenty in this class, so you should be pretty comfortable with this format whenever I talk about it. So the if portion states what the researcher is manipulating or measuring. Manipulating means change, so it's what the researcher is changing. And then it talks about the independent variable, which we'll talk about more down on the page. So that covers your if portion. It talks about what the researcher is changing. And the then portion predicts what the researcher thinks will happen. What the researcher thinks will happen. So again, you're not for sure what's going to happen. You could have a pretty darn good guess because of your prior information, because of what you researched, but you're still running the experiment because there's some doubt somewhere, okay? So it's what you think will happen. The if-then statement is the hypothesis, okay? So moving on down the page, we're going to talk about two more very important parts of the experimental design, and these are the variables, okay? And a variable in common language is anything that varies, okay? It changes, it varies, and we're going to talk about it. So the first type of variable in an experiment is your independent variable. The independent variable is the variable that the researcher establishes and can manipulate. Again, manipulate means to change. So it's the variable that the researcher, the scientist, whoever is changing. For example, you could commonly change time, or time could pass, so it's not necessarily you changing time, but time could be changing. So if you measure from a period from 1995 to 2005, time is changing. It could be the amount of water that you're adding. So you're adding something, you're changing the amount of water. You're adding nutrients, so you're changing the amount of nutrients. You could be changing the amount of light or food, or plenty of other things. We're going to talk about a lot of experimental design, lots of variables, so you're going to see a lot of different independent variables and hopefully be able to identify them pretty easily. Okay? So in an experiment, we're on this bullet, in an experiment you are only going to be changing one thing, the one independent variable. Let's highlight that. Maybe in a different color, or you're going to circle it. I don't know. But that's really important, that there's one independent variable. Okay? So in an experiment, let's say you picked a bunch of independent variables. Let's say you picked 5. You picked all of these 5 up here to change. And then you get your results. Are you going to know which of these independent variables caused that change? No, there's absolutely no way to tell which of these things changing caused that result, because so many things changed. So in an experiment, you are going to pick one independent variable that you are going to test. You have to narrow it down to one thing. And this one thing that you pick is called the experimental group, because it's the key part of the experiment. It's the one that you are changing. Okay, so moving on to the next bullet. The independent variable, called the experimental setup or experimental group, is stated in the if portion of the hypothesis. So the if portion that we talked about right up here, the first part of your hypothesis is going to be your independent variable. And we'll talk more about hypotheses and independent variables in a minute, so it'll make more sense, hopefully, if it doesn't right now. And so after you get your independent variable, you do some other things, you run the experiment, and you get your results. What we know up here, what do we do after we get our results? Do we just let them sit there and we never look at them again? No, of course not. That would be a waste of our time. So up here, from our scientific method, we know that after we do the experiment, after we collect the data, we have to analyze it. And most of the time when we analyze data, we graph it. In some way, we graph it. Okay? And when we graph it, the independent variable is plotted on the x-axis, which is the horizontal one, or the one that goes side to side. It's flat. Okay, so that sums up the independent variable. And lots of times the two types of variables can be confusing because they have the same root word. They sound a lot alike. And so the way I remember which one is which is that the independent variable starts with an I, and it is the one I, as the researcher, as a scientist, am changing, okay? So if I am changing anything in the experiment, it is going to be the independent variable. Hopefully that helps you remember it. If not, you figure out your own way, okay? So that's the independent variable, is the one that I am manipulating or changing, okay? And then we move on to the dependent variable. So it sounds a little bit like the independent variable. The dependent variable, this is the variable that the researcher is monitoring. And what does monitoring mean? It means like observing, watching, measuring. Okay? So it's the one you're monitoring. This could be many things. It could be plant height that you're measuring. It could be oxygen gas produced. It could be energy consumed that you're measuring. It could be the amount of product. It could also be the number of organisms. There are many different types of independent variables that you could be measuring. Okay, and looking at this bullet right here, the dependent variable depends on the independent variable. if you look at the root word of dependent, it's depends, and the dependent variable depends on the independent variable. So, for example, if you open a faucet, who's opening the faucet? Me, because I'm the scientist, I'm the researcher. That is my independent variable. I am changing the faucet status, okay? And what's going to happen? Water's either going to run out or water's not going to run out. But either way, we're going to measure how much water is flowing. Either a lot, a little, none, okay? And that is your dependent variable. So your dependent variable, the water flowing, you could not measure that until you open the faucet. Until you change that faucet, you can't measure anything. Okay? If you don't turn on the faucet, you're just sitting there looking at it and you can't measure anything. And that's a really boring experiment and useless. So you have to open the faucet, you have to change that, and then you will get something that you can measure. Okay? So a dependent variable basically changes in response to the independent variable. And like up here, oh, no, actually right here, the hypothesis is made up of the independent variable and the dependent variable. So the change in the dependent variable is predicted in the then portion of the hypothesis. And again, we'll take this data, we'll graph it, and we will graph it on the y-axis, or the vertical axis, the one that goes up and down. Okay, so that can be another thing that's really confusing, remembering which variable is which, but hopefully we can work that out, because independent is the one that I am changing, and the dependent depends on whatever you're changing. And then we have to deal with where we put it on the graph. But there's this pretty cool way that I learned how to remember, and that I still do remember it this way, is dry mix. So find somewhere to write this on your paper. You're writing dry mix. Somewhere where you can have plenty of room. I just don't have a lot of room on here. So this is a mnemonic device to help you remember which variable is what and which axis it goes on. Okay? So the D, hopefully you can guess that the D is for the dependent variable. And obviously don't write through your notes like this. Find somewhere else to write it. I just don't have a spare piece of paper. So the dependent variable is the response. Whoop, wrong one. The dependent variable is the response. It's what you're measuring. It is responding. It's what happens. Okay? So the dependent variable is the response, and it is graphed on the y-axis. And then we got our dependent variable taken care of. Now I have to take care of our independent variable. So that's what this I stands for. Independent. And the M stands for what the independent variable is, and it is the manipulative. Remember, manipulate means to change, so the manipulative is the one that is changing. And it is graphed on the x-axis. So I promise, make sure you wrote this down because it is very important. It's one of the easiest ways to remember how to graph things, and we'll be graphing things up until the end of the year. So very important that you know how to graph things. So continuing on with the parts of an experimental design, we just wrapped up talking about the variables, and now we're going to talk about something equally as important in the experiment. So you'll recall that I said there's only one independent variable, because if you had more than one independent variable, you wouldn't know what truly caused the results that you had. But another key thing that you need to consider is that you're comparing those results to something else. So you won't know if the results that you got were because of the independent variable or because that's what actually happens unless you have this control group. The control group is the group or setup that does not have the experimental group applied, meaning that it has none of the independent variables. There is no independent variable applied to that group. The experimenter doesn't change anything, so they're all kept the same, and the experimenter does not change anything. So they just let it run like it normally would. They're not changing anything. And in other words, the control group is used to compare the experimental data to. So if we didn't have the control group, we wouldn't know what our experimental data meant. The results would mean nothing because we had nothing to compare it to. So moving on to the final part of the experimental design that we're going to talk about, the conclusion. So the purpose of the conclusion is to analyze observations and data collected from experiment. So like I was saying earlier, you're not going to come up with a hypothesis, come up with the experimental design, run the experiment, collect all this data, and then do nothing with it. You have to analyze the data, otherwise it doesn't make any sense. So in the conclusion is where you analyze observations and data collected and study how it relates to the original hypothesis. So remember, the whole point of doing the experiment was to see if what you'd get matched what you thought you would get in your hypothesis. So you have two options in your conclusion with your hypothesis. You either accept your hypothesis if the data matched what you listed in the predicted then portion of the hypothesis. So remember, the hypothesis is made up of the if-then statement, and if what you predicted in the then portion happened, then you would accept your hypothesis because what you guessed or what you predicted would happen, it actually happened and you observed that. So if you perform an experiment and you accept your hypothesis, is it immediately going to become this great scientific theory or law, or are you immediately going to publish it? Chances are no. In order for that to happen, in order for those things to happen, it has to be retested and supported repeatedly. So things, when we talk later about laws and theories in science, it's not like one scientist conducted the experiment and got what he got and then that's what we all believe. Like, no. It happened over many, many years and many, many different people testing the same experiment and getting the same results. So hopefully when you become a scientist maybe and you are doing experiments and you get these great results, someone should take the experiment that you did and recreate it and hopefully get the same results. And it kind of validates what you were doing in your experiment. And if that happens where it's supported repeatedly, then it can be elevated to the level of law or a theory, which there are some misconceptions around and we will talk about in a minute. But there's also the chance that you do this experiment and your data does not support the hypothesis. And if that happens, are you going to give up science forever and never do anything. Never do anything with your results. No. Even if your experiment did not prove your hypothesis, that is still very valuable data and observations that you collected. So you would still say that you reject your hypothesis, but it's not a bad thing. You shouldn't be ashamed of having to reject a hypothesis because you still found valuable information. It just wasn't what you thought was going to happen. And so there are multiple options you can do if you reject your hypothesis. You're not going to give up science forever. The options are: you could redo the experiment to retest your hypothesis, see if you did something wrong or if you get the same results again, and then you truly know, okay, my hypothesis wasn't proven. So you always redo the experiment to retest the hypothesis, or you can revise, rewrite your hypothesis and run the experiment again. So if you have run the experiment and you get one thing that doesn't match your hypothesis, you can change your hypothesis to match what you got in the first experiment, and then, this is very important, you still have to run the experiment again to see if you get the same thing again. But in no part of this, if you reject your hypothesis, do you just give up. You still had very important data, and you get something from that. So that about sums up the parts of the experimental design. Whenever we run experiments, this will be the part that you end on as the conclusion. But if you look back at these steps to the scientific method, what normally happens after scientists figure out their conclusion? They have to share it with other people. So that could be their fellow peers in their same lab or in their same class, or publishing it in a journal or magazine to share it with other scientists. So it's very important to remember that the conclusion isn't the end of the experiment, but it's as far as we'll go in this class. So that kind of concludes the parts of the experimental design that we'll talk about, and now we'll get into why we do experiments, which I kind of touched on earlier, but mainly it's so you can figure out any questions that you have in science. So that's what is really driving scientists to do experiments, is because they have questions. We still don't know everything about Earth and life and how they interact, and so that's why we still do experiments. And so, like I touched on earlier, there are two major goals for scientists when they do experiments. And it's not just to get the results that they want to share those results, they want those results to be validated through multiple people doing the experiments, and hopefully it can be elevated to a law or a theory. So we're going to talk about a law first. A law in science is an explanation for patterns in data. Examples of this are the law of universal gravitation, the law of conservation of mass, which I'm sure you're familiar with, the law of thermodynamics. There are many, many laws that have been written and researched to prove these patterns in data. So we noticed things happening when we did experiments. For example, the law of conservation of mass did experiments and noticed that the mass wasn't changing, and that was their data. They noticed those patterns, and so they came up with the law of conservation of mass over years, and many different scientists contributing to it. And then the second goal for scientists could be to get a theory. Now, a theory is an explanation of events based on data. There are many theories and many misconceptions about theories, which I'll cover. And I'll remind you, in order for things to be elevated to a law or a theory, they have to be tested hundreds and hundreds and hundreds of times and proven hundreds and hundreds of times before they can get to this status. Okay? And so some examples of some common theories that we're especially going to talk about in this class are the cell theory, the genetic theory, and the theory of evolution. So, in scientific terms, a theory and a hypothesis are not the same thing. They are not the same. Put "not" in all caps. In common speech, they are used interchangeably, which leads to misuse and confusion. But in science, a theory and a hypothesis are not the same. So if you're talking to your friends and you're like, oh, I have a theory about that, that's fine for you to say in a casual conversation with your friends. But if someone says, oh, well, that's just a scientific theory, they don't really know. That's not true at all, because scientific theories, like I said, still have to be validated hundreds of times before they can become a theory. So to reiterate that point down here, a theory is not a guess, and has been validated so much that we accept it as a fact, as a scientific fact. And there is a video that we're going to watch as a class to kind of go over it even more, so you don't have to write anything for this part. But I just want you to get that a law and theory have been proven so many times that we accept them as a fact. So the big idea, the things that I want you to get out of this set of notes, is that the scientific method is an important set of steps to follow to ensure that scientists around the world are conducting experiments and gathering data in a meaningful way. So if scientists around the world are doing it, we know if they're following the scientific method, they're following the same steps that someone in another part of the world is, and that we can trust their results from their experiment. However, these steps are not solely for scientists in labs. These steps can be adapted or changed and used by anyone, making anyone who looks and thinks about a problem critically a scientist. And we will do the graph practice together as a class. So you're done with the notes for now.